many monosaccharide units join together by the glycosidic bond and they are called as polysaccharides. What are the three very important components? Sugar, 4 nitrogenous bases and phosphoric acid. The thymine of DNA is replaced by uracil of RNA. Hello everyone, welcome back to the first, that is a very important chapter called Biomolecules, that is session 4 of this chapter 9 called Biomolecules. Myself, B. Shobha Rani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. Hope you remember in the last session we studied about carbohydrates. As we know, carbohydrates, proteins and lipids are called as what? Macromolecules. As we know, carbohydrates are organic compounds made up of carbon, hydrogen and oxygen. So, as we already studied about the classification of carbohydrates. Classification of carbohydrates into monosaccharides, then oligosaccharides, then 3 polysaccharides. Polysaccharides. Monosaccharides are called single sugars. Oligosaccharides are called few sugars. Then polysaccharides are called as many sugars. So the building blocks of carbohydrates are monosaccharides. So we have already studied in the previous session about monosaccharides and oligosaccharides. Now let us study about the polysaccharides, that is the many sugars. So here we are going to study about polysaccharides. So here you can see that it is nothing but made up of many sugars. So hence the name polysaccharide. Poly means what? Many. Saccharides, monosaccharide. So many monosaccharide units join together by the glycosidic bond and they are called as polysaccharides. Now let us understand what are the, and we know the prepared food of plant, that is starch, and the cell wall of plant called cellulose are a very good classical examples for the polysaccharides. So now let us understand what are polysaccharides. So polysaccharides are long chains of sugars. So here you can see they are long chains of sugars. They are long chains of sugar. So as we know, they are nothing but organic compounds made up of carbon, hydrogen and oxygen. So polysaccharides are carbohydrates of much greater molecular weight and complexity when compared to oligosaccharides like disaccharides, trisaccharides, right? So compared to the oligosaccharides, polysaccharides are carbohydrates in much greater molecular weight and complexity. Groups are monosaccharide units joined together. Groups of monosaccharide units are joined together by glycosidic linkages. So how is a polysaccharide? The very word itself will tell you. Poly means what? Many. So many saccharide units joined together to form a polysaccharide. And what is the bond between the two monosaccharide units? It is called glycosidic bond. Glycosidic bond. So they are threads, literally a cotton thread containing different monosaccharides as building blocks. So many building blocks combine together with a glycosidic bond to form a polysaccharide. As I said, you can give a very good example is what? Starch. What is starch? The prepared food of plants through photosynthesis is called starch, and cellulose. Cellulose is the cell wall material. Cell wall material. Then even chitin. All these are nothing but polysaccharides, right? So what are polysaccharides? Polysaccharides, as we know, carbohydrates are organic compounds containing carbon, hydrogen and oxygen, and the building blocks of carbohydrates are monosaccharides. So what is the classification of carbohydrates? Carbohydrates have been broadly classified into monosaccharides, that is single sugars, then oligosaccharides called few sugars like disaccharides, then polysaccharides called many sugars. So polysaccharides are what? Long chains of sugars, and they are more higher in molecular weight and complexity when compared to oligosaccharides, and groups of monosaccharide units are joined together by glycosidic linkages. So they contain many monosaccharide units joined by the glycosidic bond. And what are the building blocks of polysaccharides? Monosaccharides are the single sugars are the building blocks of polysaccharides. Polysaccharides are classified into, now let us move on to the classification. They have been broadly classified into homopolysaccharides and heteropolysaccharides. So we have understood what is polysaccharides. They are nothing but long chains of sugars which have been again broadly classified into homopolysaccharides or heteropolysaccharides. You can also use the word called homopolymers or heteropolymers. Now, what is homopolysaccharide? The very word itself will tell you. Homo means what? Same. What do you mean by hetero? Hetero means what? Different. So, what are homopolysaccharides? They are the polysaccharides containing only a single type of monosaccharides. So, they all contain only one type of monosaccharides, and such polysaccharides are called as homopolysaccharide. For example, you can take example of starch, glycogen and cellulose. What are hetero? The very word itself will tell you. They are the polysaccharides containing two or more different kinds of monosaccharide units, hence the name. They are called as heteropolysaccharides. So bacterial, cell envelope, chitin, etc. All these are the classical examples for heteropolysaccharides. So what are polysaccharides? They are a type of carbohydrates which are long chains of sugars. Then they are more in molecular weight and complexity, and they are joined, and many monosaccharide units are joined together by the glycosidic linkages. And the building blocks of polysaccharides are the monosaccharides, and they have been broadly classified into what? Homopolysaccharides and heteropolysaccharides based on the type of monosaccharide units involved. And homopolysaccharides are those polysaccharides which contain a single type of monosaccharides. For example, saccharides, for example, starch, glycogen and cellulose. Heteropolysaccharides, which are also called homopolysaccharides, are also called as homopolymers. Heteropolysaccharides are also called as heteropolymers. Then containing two or more different kinds of monosaccharide units, and the classical example you can give is bacterial cell envelope, chitin, etc. So here you can see starch, cellulose, glycogen. All these are the examples of polysaccharides. Cellulose, I think all of us know how important, as we know that we know the difference between a plant cell and an animal cell. So the first very unique difference between a plant cell and an animal cell is what? Plant cell have the cell wall material, outermost cell wall material called cellulose. So here you can see the beautiful picture of a leaf. So cellulose means cell wall of plant cell is made up of cellulose, which is nothing but a polysaccharide. So I think all of us know the plant cell wall is more rigid and tough. And this rigidity and toughness of the plant cell wall is mainly due to the presence of the primary cell wall material called cellulose. So a fibrous, tough water insoluble substance is the chief constituent of cell wall of plants. So what is cellulose? It is a fibrous, tough, water insoluble substance, and it is the chief constituent of the cell wall of plants. And cotton fiber, I think all of us know cotton has wide economic importance, which is used in the process of making clothes. So it is nothing but a classical example for cellulose. Then plant cell walls are made up of cellulose, then made up from, I think all of you know paper. Paper is made from plant pulp is called cellulose. So it is made from what? Plant pulp. I think all of us know paper. Plants are used for making of paper. So what is that which is prepared? How is paper made? From the, that is the pulp. Plant pulp is used for making paper, and plant pulp contains nothing but cellulose. So cellulose is a very important polysaccharide. A very good example for polysaccharide. So what is cellulose? It is a fibrous, tough. It is one of the example for polysaccharide. And as we know, plant cell wall is very rigid and very tough, and the rigidity and the toughness of the plant cell wall is mainly due to the presence of the cellulose, and it is a fibrous tough water insoluble substance which forms a very important constituent of the cell wall of plants. And cotton fiber is cellulose. Then plant cell walls are made up of cellulose, and paper from plant pulp is cellulose. As we know, it is used for making of paper. Then starch. So as we know, the prepared food of plant is called as starch. So all these are the examples for the food that is stored with starch. I think all of us know potato. You know, potato is one such vegetable which is quite commonly used in making most of the dishes. And it is because of the stickiness which acts as a binding agent. And that is because of the starch. Rice will have more of starch, the stickiness. And that stickiness itself is nothing but for the presence of starch. So now let us understand what is starch. See how beautiful this picture is, which gives us an idea about the sources of starch. Sources of starch. So starch, it is the important storage polysaccharide in plant cells. It is a very important storage polysaccharide, and glycogen is abundant in liver. Even glycogen, as we know, it is also one of the storage food. Then inulin, it is a natural storage polysaccharide found in wheat, onion and bananas. So the inulin belongs to a class of dietary fibers known as fructans, and it is a polymer of fructose. Then there are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars. Example, like we have glucosamine, N-acetyl galactosamine, etc. Then exoskeleton of arthropods. I think all of us know what is a exoskeleton. The skeletal structure that forms outside the body of the arthropods, particularly insects, have a complex polysaccharide called chitin. So imagine the insects are able to survive in almost all climatic conditions. It is because of their protective exoskeleton, which is made up of chitin. So what are the other examples for polysaccharides? One is starch. Starch, as we know, it is a very important storage polysaccharide in plants, and the prepared food of plants is nothing but starch. Then glycogen is another example, which is again abundant, which is stored in the liver, and whenever it is, then sometimes glycogen is converted back into glucose. Glucose is converted back into glycogen to maintain the glucose level in the blood. Then inulin, which is nothing but a natural storage polysaccharide found in wheat. Here you can see the wheat, then in onion, then bananas. Then the inulins belong to a class of dietary fibers known as fructans. It is a polymer of fructose. There are many more complex polysaccharides in nature which acts as a building blocks. Example, you can take glucosamine, N-acetyl galactosamine, etc. Then exoskeleton of arthropods, for example, have a complex polysaccharide called chitin. All these are the different examples for polysaccharides. Moving on to the next very important part of this session about biomolecules is nucleic acids. They play a very great role in both the plant body and the animal body. So what are nucleic acids? How come the name nucleic acid has come? These nucleic acids, namely the two nucleic acids, are DNA and RNA. Because these are called nucleic acid, they are found in the nucleus of a cell, found in the nucleus of a cell, and show acidic property, hence the name nucleic acid. So why they are called as DNA and RNA are called as nucleic acids? What is DNA? Deoxyribose nucleic acid. What is RNA? Ribose nucleic acid. Here you can see DNA and RNA. So as we know, DNA is a double-stranded structure, RNA is a single-stranded structure. DNA is a genetic material in all the organisms, whereas RNA exists in two forms as genetic RNA and non-genetic RNA, right? So why are they named as nucleic acids? Because they are found in the nucleus of a cell and show acidic property, hence the name nucleic acid. So what are the two types of nucleic acid? DNA and RNA. Now let us understand. as I said, DNA is the genetic material. As we know, what is gene? Gene is nothing but DNA. DNA is nothing but gene. As we know, DNA is always double-stranded. It never exists as single-stranded, whereas RNA exists in both forms. So it acts as genetic RNA in some of the prokaryotes, and it is very important in protein synthesis. So when it is involved in the process of protein making, it is single-stranded. So when it is acting as a genetic material, it will be double-stranded. So now let us understand what is this nucleic acid. And the word nuclein was coined by Friedrich Mischer. So when Friedrich Mischer subjected the hospital bandage pus cells with certain digestive enzymes, one part of the cell remained undigested, and he named that undigested part of the cell as nuclein. And that is how the word nucleic has come into existence. So the word nuclein was coined by Friedrich Mischer, and this scientist called Friedrich Mischer subjected the hospital bandage cells with certain digestive enzymes, and one part of the cell remained undigested, and that undigested part of the cell he called it as nuclein, and that is how, and they show acidic property, hence they were called as nucleic acids. So what are nucleic acids? A complex organic compounds in living cells that of a chain of nucleotides is called, they are made up of what? A chain of nucleotides. Chain of nucleotides. So we use the word called polynucleotides. Polynucleotides. So it is double-stranded. Here you can see it is double-stranded. Here it is single-stranded. Single-stranded polynucleotides. Here it is double-stranded polynucleotides. So a complex organic compound in living cells that is made up of a chain of nucleotides. And what are the two types of nucleic acid? One is called DNA deoxyribose nucleic acid and RNA ribonucleic acid. So here it is made up of, both are made up of 5 carbon sugars. Right? Nucleotide. What do you mean by nucleotide? Nucleotide has 3 chemicals distinct components, chemically distinct components. So nucleotide has 3 very important chemically distinct components. One is called nitrogenous base, that is purines and pyrimidines, and a pentose sugar and a phosphate group. So what is a very important 3 components? What are the 3 very important composition, or you can also use the word called chemical composition? One is the 4 nitrogenous bases. 4 nitrogenous bases. 4 nitrogenous bases are nothing but amino acids. They are nothing but amino acids. Then sugar. First is sugar. Then third one is phosphate group. Only when phosphate group is added, then only it will be called as what? Acid. So now let us understand how the DNA and RNA differs with reference to chemical composition. Nitrogenous base is a heterocyclic compound. It is a heterocyclic compound. Pentose sugar is a monosaccharide. Phosphoric acid. Together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or a cell. The nitrogenous bases fall into 2 types. That is purines and pyrimidines. Purines are double ring structures. Pyrimidines are single benzene ring structures. So with reference to DNA and RNA, 4 nitrogenous, first sugar. Here it is 5 carbon sugar called deoxyribose sugar. Here it is made up of ribose sugar. So here one oxygen atom is less. One oxygen atom is less. So this is first composition. Then 4 nitrogenous bases. 4 nitrogenous bases. So what are the 4 nitrogenous bases? adenine, guanine, cytosine and thymine. Here what are the 4 nitrogenous bases? Here it is, what are the 4 nitrogenous bases? Here it is adenine, guanine, cytosine and uracil. See, here it is uracil, here it is thymine. So with reference to nitrogenous base, what are the very 2 important differences between adenine, differences between DNA and RNA? The 4 nitrogenous bases found in DNA is adenine, guanine, cytosine and thymine, whereas in RNA it is adenine, guanine, cytosine and uracil. Here it is deoxyribose sugar and here it is ribose sugar. And because both we use the word called acid, so here also phosphoric acid present, that is H3PO4, H3PO4, here also H3PO4. So this is about the chemical composition. So with respect to chemical composition, what are the 3 very important components? Sugar, 4 nitrogenous bases and phosphoric acid. So with reference to sugar, what is the type of sugar found in DNA? Deoxyribose sugar. What is the type of sugar found in RNA? Ribose sugar. So the very difference is one oxygen atom is less in DNA, whereas one oxygen atom is more in RNA. Then what are the 4 nitrogenous bases found in DNA? What are nitrogenous bases? They are nothing but amino acids. As we all know, there are 20 different amino acids. So adenine, guanine, cytosine and thymine. Here, what are the 4 nitrogenous bases found in RNA? Adenine, guanine, cytosine and uracil. What is the 3rd chemical composition? It is that is the presence of phosphoric acid in DNA also as well as in RNA. And DNA is a genetic material. RNA acts as both genetic material as well as non-genetic material. Then DNA is always double-stranded, but RNA is single-stranded. But when it is acting as a genetic material, it will also become double-stranded. So all these are the very important differences between DNA and RNA with respect to chemical composition. Then what are pyrimidines and purines? So pyrimidines are, it has cytosine, uracil and thymine. Purines, it has adenine and guanine. So right, now each nucleic acid contains what? 4 nitrogenous bases. DNA has what? ADGC, while RNA has AUGC. So DNA will have adenine, guanine, cytosine and thymine, whereas RNA will have adenine, guanine, cytosine and uracil. So DNA, what are the 4 nitrogenous bases found in DNA? Adenine, guanine, cytosine and thymine. Here, what are the 4 nitrogenous bases found in RNA? Adenine, guanine, cytosine and uracil. So here, the thymine of DNA is replaced by uracil of RNA. So what is the significance of nucleic acid? As we all know, DNA acts as a genetic material. And why it is double-stranded? Because DNA, one of the parent, as we know, it is very much responsible for the transfer of characters from the parents to the children. Hence, it acts as a genetic material. So one of the parental strand is retained in the daughter cell, and that is how the characters are transferred from the parents to the children. So it makes a replica of itself at the time of cell division, what we call it as DNA replication. So DNA acts as a genetic material, but as we know, RNA also acts as a genetic material, but it acts as non-genetic RNA, which is very much involved in the process of protein synthesis. So DNA determines the structure, function and biological characteristics of an individual and controls all cellular activities. Right? Whereas RNA acts as a genetic material in some viruses, and in higher organisms, as I said, RNA play an important role in protein synthesis. Hope you all have understood all the concepts what I have explained in this particular session. So I'll be back in the coming session with some more interesting concepts of this chapter called Biomolecules. Till then, goodbye and thank you.